Stolz, David? Okay, I'm very happy that everybody's here. So numerous, so many people. It's a hot topic, and it's really cool to see that the young people are interested in engaging in a discussion about the technology and also about the consequences, implications for our society. Before we start with the technology talk, I give you a quick introduction what we do at the Founders Club. And I will take you on a tour of what we will do in the summer term. But before this, I want to thank Pia Decker. That's a startup based in Freiburg. And yeah, right now she's drinking it. You can show it. Yeah, it's actually not a brewery itself. It's a brewery collective. That means they foster like very small breweries which are close. Wahrscheinlich die Batterie. Oder du nimmst das mit dem Kabel dran. Ja, es funktioniert wieder. Yeah, it's a collective which is like empowering like very small breweries which would die otherwise. And they have like a common marketing strategy, and they facilitate like the sales. So thanks to them, we have like some beer for free tonight. And then a big thanks to the Gründerbüro, which supported us in preparing and organizing this event. And now we will see. Next week we will have a trip to Mannheim, and it's a student congress organized by our partners, which are called Q Summit. And it's going to be like 3 days with different workshops with speakers which talk about the future of education or of bioscience. It's a really diverse set of topics, and it's also for us a great possibility to enhance our team building, to get to know each other better. Then we have somebody from a very successful e-commerce startup. It's called Emma, and they are trying to attack Casper, which is a silicon-based company, and they want to pivot the market from mattresses. Because what has been the strategy in the past 50 years, you sell a mattress with a discount of 70%, and everybody knows it's bullshit. They just make an arbitrary price, they deduct 70%, and that's their sales strategy. So Emma has a very transparent listing of the supply chain, what is transportation, what is marketing, what is manufacturing, and they see a tremendous growth, and it's very dynamic. And the founders told me, what is key in this part is the customer journey, because you're not in the store with the people, you have to chat with them, you have to Skype with them, and so you have to be very sensitive and very sensible what people say, what they write. And he's going to talk about that. Everyone who wants to build up an e-commerce empire is a very good offer. Then we have a typical Freiburg story. And it started as a research group at the Fraunhofer Institute, and they became friends, they became a very close group, and also they found something which was able to get to the market, and they actually installed a software and a hardware device into manufacturing companies, and they are regulating and optimizing the energy grid within the company, and they managed up to 7%, 10% savings as soon as it is installed. There is no latency. That's very interesting. And then for us, also a very important social aspect, we have a parent organization which is called Gründermagnet. They are based in Berlin, and the initiatives are spread all over Austria, Switzerland and Germany. And once in the year we gather in one city. This time it's Münster. And also, for this purpose, we will have a nice trip to another city and get to know other students. In June, we have the Freiburg Hackathon. It's the first time that we are co-organizing this event, so we hope it will be even better than last year. And the registration is open. So when you're a developer, of course, apply. If you are creative, if you have an idea, if you have the will to improve the living in Freiburg, come and join. And what is also part of our philosophy, we want to educate and be instructive to the members, so they have also the chance to develop themselves in the future. And design thinking is a technique which is used to to evaluate different ideas. So it works like also with like the game that you try to impose yourself in a different role, and especially into the client. And it has also like parts from theater and dramaturgy. Then those are candidate events because they are not confirmed yet, but we are optimistic that we can realize at least 2 of them. Who of you knows Rocket Internet? Yeah, there are some people. So it's like they are like the company behind Zalando and many other startups in Germany. They are actually like the biggest VC fund in Germany, and yeah, we want like to bring them here and that they can share their knowledge. What do you have to do in the first 100 days if you have a company? Then Digitalkasten, they actually want to make rid of like the regular analog cost. So this is very interesting when you're traveling, because like they have an ORC technique which recognizes every letter semantically, and so like the PDF document that you get instead of your mail is searchable. You can like also like categorize it automatically, and it will sooner or later like replace our Office box. And then what is like besides like CRISPR-Cas9, something which is really like a topic which is like very important to us is like the peer-to-peer technology, also known as blockchain. But it's way more than Bitcoin. It's like enabling each of one of us like that we regain ownership of our own data. So this is very abstract now because we cannot engage now, but we are in discussion with some experts here in Freiburg and we want to realize something. And to sum it up, we met Daniel Jung at a congress and we were talking about also the possibility to cooperate, and when we are lucky, he will come here and have a presentation about the future of education. And if you want to start straight away something and you want to have support, then I can really recommend you something from a dear friend who's leading like the startup Hatter Camp. He is Dominic. Dominic, is he here? I don't know. But if you want to have like more information about this, he's a very kind person. You can reach out to him anytime. But the cool thing is, even if you don't have like a prototype or if you have nothing, you're invited to send your application. It's not formal, it can be very short. And then it will be like, for the summer term, there will be 6 sessions, which will last 3 hours. And then you will go from like generating ideas, evaluating ideas, working on services and products, to then pitching in front of investors. And at the same time, gaining ECTS points. So the time that you invest is also like recompensated at the end. And now we're at the next slide. It would be cool and very, very great of you if you can support us and give us a like. It really helps us to realize such projects in the future, and it's also for you the best way to stay informed, because we gather all relevant information about the startup, the startup scene in Freiburg, and of course we also like have a regular update of our own events. And if you say, I want to do more, I really want to be part of the club, then you're also happily invited to just drop a message to Felix, quickly specify your subject of study, and then we will come back to you. But also for all relevant information, you see like the people with the white shirt and the buttons here, you can just go and talk to them, and we will answer all your questions. Okay, now I will hand over to my colleague, Head of Finance, Marcel, which is going to introduce our star guest, Martin Jinek.
So, it's a great honor for the Foundersclub Freiburg today to have Professor Jinek from the University of Zurich here in Freiburg. Professor Jinek studied chemistry and natural sciences in Cambridge at the Trinity College. Then he did his PhD in Heidelberg with in the working group of Professor Elena Conti. And then he moved to the University of California in Berkeley, where he did his postdoctoral studies in the group of Jennifer Doudna. And at this time, 2012, there was a paper published that is called A Programmable Dual RNA-Guided DNA Endonucleases and Adaptive Bacterial Immunity. And Professor Jinek was the first author of this paper, and this paper is kind of the basis of the whole CRISPR-Cas story that evolved from that. And it was also declared by Science, by the journal Science, to be the breakthrough of the year 2015, so the CRISPR-Cas method. And I don't want to talk much about it because I think it will suck in comparison to what we will hear right now. And just saying thank you to be here, and we are all seriously happy to have you here and interested to hear what you have to say. Thank you.
We'll start by thanking people at Foundersclub for their kind invitation to come here to Freiburg to give you an overview of some of the science behind the technology, but also the developments of this technology towards applications. Can you hear me in the back, by the way? Is it okay? Yeah?
The second microphone.
Okay.
Right.
This work? Can you hear me? Okay. All right. I'll have to handle two things at the same time. This might be a bit difficult, but bear with me. Okay, so thank you very much for the invitation. It's really great to see so many people here, and I'll try not to disappoint. So just kind of by way of introducing myself, I'm now an associate professor at the University of Zurich in the Department of Biochemistry. I've been there for just over 5 years, and in that time I've basically built up a really fantastic group of young people who have kind of joined me and worked with me on various scientific projects. We're located in Zurich at the Erkel campus, and this is kind of the view that you get of the Alps on a good day. And this is basically all the different foundations and agencies that pay our bills. So this is where our research funding comes from. So in my spare time, since this is a founders club, I'm also a co-founder of a company, and this is a company called Caribou Biosciences, based in Berkeley, California, that's focusing on the development of next generation genome editing technologies and their applications in various areas, primarily therapeutics, but also other potential uses. And just to kind of showcase that you can really go from a PhD to a CEO without anything in between. This is the current CEO of the company, Rachel Horowitz, who was a PhD student in the same lab where I was in Jennifer Doudna's group in Berkeley. So it's a really success story, and I'll spend a little bit of time talking about this towards the end of my presentation. So you may have heard about a thing called CRISPR, and that somehow this is really. I hope that this is. Okay. I think it's just the cable moving, so maybe I'll just shout. Okay. So I'll shout. So you may have heard about CRISPR-Cas genome editing, and that this is really a revolutionary technology that's very rapidly changing not just basic research but also applications in biotechnology and in molecular medicine. The reason why it's so revolutionary is because, above all, it's simple, it's fast, it's precise, and compared to some of the previous technologies, it's actually very low cost. And so, thanks to these technologies, I think in the next few years we will see genetic therapies really becoming a reality. And this also brings up another important issue in that this technology gives us the power to modify our own DNA, to modify the genome of humans in a way that could potentially be passed on to the next generation. And with that comes a whole host of questions about ethics, about responsible use of this technology, and so on. Okay, so my story goes back to the time when I was a postdoctoral researcher in Berkeley in California, in the group of Jennifer Doudna. And as Marcel already pointed out, in 2012, through a collaborative project with Emmanuel Charpentier's group and all the other people here, Christophe, Michael, Ines, primarily Christophe, we discovered that in a bacterial immune system there is such a thing called Cas9, a protein that basically functions as a DNA cutting enzyme that can be programmed. And the kind of the molecular principle behind this is that it's a protein molecule whose job it is to cut a double-stranded piece of DNA. And to basically know where to cut the DNA, the protein associates basically with a short RNA molecule that tells the protein where in the DNA it cuts by providing sequence specificity, by essentially using base-pairing interactions with one of the strands of the DNA to bring the enzyme to the right place in the DNA to be able to cut it. Now, in that same study, we were able to show that we could in fact re-engineer the system, and unlike the natural system, which uses two RNA molecules and one protein, we developed a kind of a simplified version of it that basically consists of just one protein and one RNA molecule. And this is essentially the basis of the technology as we know it now. Basically, all you need to do to be able to cut a piece of DNA that has a specific sequence is to design an RNA molecule that will base pair to one of the strands in the DNA at that specific site, and you have to combine it with the Cas9 protein, which then does the job of catalyzing the chemistry of DNA hydrolysis, so the DNA cutting reaction. And so the power of this technology comes from the fact that you can take the system out of bacteria and you can introduce it into other cells. You can introduce it into animal cells, plant cells. You could introduce it into human cells. And if you program it with a specific RNA that targets a sequence in the genome of that cell, you can basically break that DNA into two pieces. Now this is something that cell senses as damage and will try to repair it using DNA repair pathways. However, these DNA repair pathways can be basically exploited and to some extent controlled so that the way that break is repaired when the DNA is chemically ligated back together results in some modifications to the sequence of the DNA. And then basically depending on which DNA repair pathway it is, you can come out with different types of modifications. But the bottom line is that in order to do that, first you have to cut the DNA and then you have to let the cell repair the DNA, which then leads to the modification. The details are not so important for the purpose of this talk, but nowadays with this technology you can really do everything. You can insert letters into the genetic code, you can delete letters from the genetic code, you can overwrite parts of the genetic sequence. So on some level, this is really like working with a text editor on a computer. You design basically your guide RNA, and then you let the system basically do its job. And so, kind of following up on that initial discovery, which was focused on the applications in vitro, we were also one of the first groups amongst these five to show that you could in fact apply this system in other cells than bacteria, so in human cells, in cultured human cells, and we could get genetic modifications in these systems. And so since then, in less than 5 years, we've seen that basically this is now really revolutionizing many different areas. So of course it's become a really powerful research tool that allows us to study the functions of genes in many different organisms, many different types of cells and so on. But it's rapidly changing the face of biotechnology and above all molecular medicine. And the hope is that very soon, in not so distant future, we will see human therapies based on based on this technology. Now, the main kind of selling point of this technology is that it's essentially brought down the cost of doing genome editing on a per gene basis. If you just count the cost of the reagents that you would need to have in place to modify a gene in a cell by about 2 orders of magnitude. So some of the previous technologies like TALENs were very cumbersome to apply. Basically, it takes a long time to generate these reagents or to design them in the first place. Whereas in the case of CRISPR-Cas, since all you need to do to target it to a specific genetic sequence is to design a piece of RNA that you can synthesize quite cheaply these days, that basically brings down the cost quite substantially. And this has really brought the genome editing technology to the masses, so to speak. Not just in research, but also for applications. So what I'll do in kind of little 4 points throughout my presentation is to give you an overview of where the CRISPR-Cas system comes from, then tell you a little bit how these genome editors actually work, how we can implement them practically, and then finally how we can use these technologies for genetic therapies. And this is kind of a more forward-looking end. And so the system, as I already alluded to, comes from bacteria. And it comes from a kind of a genetic defense mechanism that allows bacteria, and by the way, this is a bacterium as imaged by an electron microscope, that allows bacteria to defend themselves against viruses. So just like humans and plants and animals, bacteria also get viral infections. And since this has been going on for quite some time in evolution, they've actually evolved pretty sophisticated mechanisms to defend themselves against these viruses. And so the CRISPR-Cas system is essentially, in its naturally occurring form, a genetic defense mechanism against viruses. Now, to tell you a little bit about this, I will play you a short video that was put together by the Max Planck Society. And is available on YouTube and other websites. So I hope the sound works.
Viren sind eine Bedrohung für alle Organismen, selbst für Bakterien. Sie docken an die Zellen an und schleusen ihre DNA ein. Diese wird in das Bakterienerbgut eingebaut und steuert so die Produktion neuer Viren. Doch die Bakterien sind nicht schutzlos. Sie prägen sich den Erreger ein und können deshalb eine Immunität gegen dieses bestimmte Virus entwickeln, indem Enzyme ein Stück aus der Viren-DNA herausschneiden und es an einer ganz bestimmten Stelle im Bakterienerbgut einbauen. CRISPR-Abschnitt genannt. Die Zelle übersetzt diesen dann in ein RNA-Molekül, die CRISPR-RNA. Sie enthält nun Informationen aus dem Erbgut des Virus und des Bakteriums. Jetzt kommt ein zweites Molekül ins Spiel, die Tracer-RNA. Gemeinsam binden sie an das Enzym Cas9. Dieses Enzym schützt nun gegen genau dieses Virus, denn seine CRISPR-RNA kann an die entsprechende Virus-DNA binden. Das Cas9-Protein zerschneidet daraufhin den DNA-Strang und macht ihn so unschädlich. Der Angriff des Virus ist dadurch abgewehrt. Die CRISPR-RNA bestimmt also, wo Cas9 die Viren-DNA schneidet. Doch das funktioniert nur, wenn auch die Tracer-RNA gebunden ist. Zwei Entdeckungen machen das CRISPR-Cas9-System nun zu einem revolutionären Werkzeug für die Gentechnik. Die Tracer- und CRISPR-RNA lassen sich zu einem einzigen Molekül fusionieren. Dadurch können Wissenschaftler sie leichter herstellen. Und mit einem modifizierten Cas9-Protein funktioniert das System auch in höheren Organismen, die Zellen mit Zellkern besitzen. Forscher können die Sequenz der fusionierten RNA-Moleküle problemlos variieren. Dadurch bestimmen sie, wo das Cas9-Protein einen DNA-Strang zerschneidet. So können sie beliebige Gene ganz gezielt ausschalten. Die Zelle versucht zwar, die losen Enden wieder zusammenzufügen, Doch das gelingt ihr nicht fehlerfrei, weshalb das Gen nicht mehr abgelesen werden kann. Es können auch komplette Gene ausgetauscht werden. Diese Methode ist wesentlich preiswerter und effizienter als alle bisher bekannten Techniken, denn es muss nur ein RNA-Molekül verändert werden. Mit CRISPR-Cas9 lässt sich das Erbgut jeder beliebigen Zelle verändern. Eines Tages könnte dieses präzise Werkzeug vielleicht sogar helfen, Erbkrankheiten beim Menschen zu heilen.
All right, so, all right.
On?
Hello? Yeah. Good. Okay. So I hope that this gave you at least a little bit of a flavor for how this system actually came about. And I should point out that this really was thanks to basic research. That this is not something that was really kind of designed or discovered with the purpose of doing genome editing in the first place, but really came through just basic curiosity in the way bacteria function and how in the way nucleic acids function in cells. And so the next thing I'll tell you a little bit about is how these genome editors actually work. And this again goes back to this idea that we have a protein molecule that binds a guide RNA that allows it to then target a specific sequence in a double-stranded DNA molecule. And again, we can basically simplify the system using this trick of fusing the two RNAs into one. So in my lab in Zurich, we're really focused at understanding the basic molecular mechanisms behind these technologies. And we do this by studying the three-dimensional structures of the molecules that are involved. And this is an example of one of such structures that we produced in the lab. This is a 3D printed model, but it's based on basically data that we generated by X-ray crystallography, by one of the structural techniques that are commonly used in biochemistry. And basically, it's a model that shows you quite nicely how tightly everything fits together in these molecules. Okay, that in white we have the Cas9 enzyme, and then in red we have the guide RNA, and in blue is one of the DNA strands that basically wraps around the RNA, forms a double helix with the RNA, and this is basically how the enzyme can find the right place in the DNA to be able to cut it. We can also really study this at atomic resolution, so we have basically an idea about individual interactions between atoms. So here we have, this is a part of the protein that recognizes specific bases in the DNA, and this is important for the initial binding mechanism for the targeting process. As it turns out, there's not just Cas9, there are other now CRISPR-associated enzymes that have been repurposed for genome editing. Another one of these is called Cas12a. And again, this is an RNA-guided enzyme that has some interesting differences to the Cas9 system. So in this case, it is naturally guided by just one RNA molecule. It doesn't need the second one. It cuts the DNA in a slightly different way. So unlike Cas9, which basically makes essentially a blunt end break, this makes a break that has staggered ends, and this again can be used for applications. And so in my lab, we've also very recently started studying the three-dimensional structures of these molecules. And the hope is really that we can use these molecular insights to improve the properties of these technologies. Even though these technologies are really quite specific and they are very versatile, there's still quite some room for improvement, and we're hoping that by exploiting the molecular insights, we can contribute to structure-guided engineering of these enzymes to make specific substitutions of amino acids and so on that can then improve the properties, to improve the specificity, to make these enzymes more efficient in specific cell types, or to be able to somehow influence the way the DNA cutting then leads to specific DNA repair outcomes. So this is kind of the technology-focused aspect of our work. Now, how can we actually practically implement CRISPR-Cas9 genome editing? So I already told you it's all about being able to make that double-strand break in the DNA. And this is basically by essentially introducing both Cas9 and the guide RNA into the cell where you want to do the editing. And so if that can be done, then you end up with a double-strand break, and that, as I said, is sensed as damage by the cell. Cells don't like to have breaks in their DNA, and they have evolved specific mechanisms to repair these breaks. Now, depending on which DNA repair pathway it is, and there are several that can do this in parallel, you can have different outcomes. And so one of the pathways, it's called non-homologous end joining. This is it's not really important, but this basically is a kind of a quick and dirty mechanism that the cell uses to really quickly put the two ends back together. One of its features is that it's not a very precise mechanism, and so very often when the two ends are put back together, there will be random insertions or deletions at the breakpoint. So 1 or 2 or 3 of the letters will be lost, or some random ones will be added at the breakpoint. And so this is a way you can actually knock out a gene. If you basically target the middle, the coding sequence of a gene, you can basically inactivate it in this way. So this is very good for breaking things. If you want to actually do things in a more precise way, what you need to do is to exploit a different kind of DNA repair pathway in which the break in the DNA is repaired by using information that comes from the second copy of that sequence. And this can be either from the DNA that has been freshly replicated, or from the second chromosome, that basically the second copy of the chromosome that normal diploid cells have. And so what happens here is that the cell basically uses that other DNA as a template. And so what you can do is basically trick the cell into using something that you provide exogenously. So simultaneously with providing the protein and the RNA to target the gene, you also provide a piece of DNA that the cell then takes as the repair template and will repair the break using that. And that basically then can lead to, depending on how this template is designed, to a specific modification. So you can either insert a sequence or overwrite a piece of a sequence, or you can also delete a piece of genetic sequence in a specific place. And so this DNA repair pathway, known as homology-directed repair or homologous recombination, is basically what allows you to do specific modifications really with single letter precision. So you can effectively do gene surgery, really very, very targeted interventions in the genome. Now, in order to be able to do that, however, you have to be able to deliver the Cas9 protein and the guide RNA into cells. And there are various ways of doing that. You can, for example, use lipid nanoparticles or engineered viruses to basically deliver DNA that encodes these components. So basically a piece of DNA that when introduced into the cell will be transcribed and then translated to make a Cas9 protein. The guide RNA will be also transcribed from the DNA, and that's one way of doing it. In other systems, you can basically deliver these two components as RNA. So in this case, the Cas9 will be translated to make a protein. This is something that you can typically do with if you do editing in organisms that have cells that are large enough to be injected, because this is something that you can do quite easily. So if you work, let's say, with a model system like zebrafish or Drosophila, where the cells that you want to target are essentially, visible under almost with naked eye, you can basically you can deliver these components just by simply injecting the cells. In other systems, you can also basically take a purified Cas9 protein and just basically mix it with an RNA in a test tube and then deliver it into cells either again by microinjection or by giving the cells an electric shock, something known as electroporation or nucleofection. And it's something that's becoming more and more commonly used in applications. So you have a variety of choices, and it really depends on which organism you work with or which cell type you work with, because some of these methods work better than others in specific cases. There are other variations on the theme. You can now also do genome editing without actually having to cut the DNA in the first place, and this is basically because you can engineer the Cas9 protein to basically retain the DNA binding mechanism that's dependent on the guide RNA, but not actually be able to cut the DNA. And you can use it as a kind of a delivery platform to bring other proteins that are fused to the Cas9 protein to the DNA and let those then do something to the DNA. And so in this case, you basically bring in DNA modification enzymes that can basically turn one type of base into another and do editing with, again, quite a high degree of precision without actually having to cut the DNA in the first place. So that's another kind of twist on the basic technology that has emerged in the last couple of years. And so finally, let me talk a little bit about how we can use these technologies for genetic therapies. Now, the idea is that unlike previously, when someone has a genetic defect, so let's say a point mutation in one of the globin genes that basically causes sickle cell anemia, the only way previously to cure this genetically would be to provide another copy of the gene and sort of rely on this copy being inserted somewhere in the genome, which is something that could not be done very, very controllably. And so where CRISPR-Cas genome editing could play a major role in the not-so-distant future is basically gene therapy 2.0, and that's the idea of essentially going back to exactly the place where the problem is, so back to the gene that contains the mutation, and using CRISPR-Cas to basically repair that mutation back to wild type, okay, back to the healthy, normal functioning gene. So you could potentially basically go from repairing that mutation to a healthy individual. And there would be potentially two different ways of doing it. One would be to essentially deliver the components of the CRISPR-Cas technology into the patient, and that basically means having some kind of a delivery system that would be specific for a specific organ or a tissue or a cell type. And this is something that is still currently a major challenge. The other possibility is to do the genome editing actually ex vivo, so outside of the body of the patient. You would, of course, this would not most likely be applicable to all diseases, but you could envision basically extracting cells containing that genetic defect from the patient, and then making the modification in cell culture, basically in a dish in a lab, and then returning the cells back into the patient to have some curative outcome. You could even think about taking it one step further, namely that you could take cells, let's say fibroblasts, so from skin cells from a patient, and turn them into stem cells in the lab by reprogramming, and then modify these stem cells, differentiating them into specific cell type. Let's say epithelial cells for the gut or liver cells or whatever, and then introducing them back into the patient. And so this is one of the major areas that both academic as well as industrial players in the genome editing space are focusing on. Basically, this type of ex vivo approach is most likely going to be the one that will first yield some concrete results. And it's really looking quite good. I mean, there have been now quite a number of studies showing that, at least for various model systems that are either cell-based or even in some organisms, you can basically practice this approach. You can isolate cells, you can modify them, and you can return them and have a curative effect. You can also there has been now quite a number of studies showing that these in vivo approaches, if you have a good delivery system, can work. An example of this was basically these 3 studies that came out in 2016 showing that you can treat muscular dystrophy in mice by basically delivering these CRISPR-Cas9 components using a virus. Basically, by infecting cells with a modified virus that carries the Cas9 protein and the guide RNA, you can achieve, at least in an animal model, basically a substantial curative effect, basically. Things have gone as far as basically they're now being the first clinical trials using CRISPR-Cas genome editing approved. And this is focusing on the field of immunotherapy against cancer, so modifying basically immune cells so that they can target cancer cells in patients. And it's becoming clear that CRISPR-Cas9 genome editing will play an important role basically in this. So now there's quite a few. These were the first 2, but there are several more now that have been approved by the regulatory agencies, both in the US and in China, that will basically be doing this. But to really make this work efficiently and safely and be widely applicable, there are still quite some technical challenges that I think will have to be worked out in the near future. One key question is the specificity of the technology and how to really avoid off-target effects. So basically, the potential for the system to have a side effect and modifying some other part of the genome that was not intended to be modified. Of course, the issue of delivery to specific cells or specific organs is something that is an ongoing issue, and there's a lot of effort going into devising new delivery methods. And then, of course, this idea of being able to control the DNA repair outcomes better and more efficiently. And this is, for example, again, where using some of these alternative nucleases or this base editing technology could also play a role. So finally, I'll say a little bit about the company that I've had the fortune of being able to co-found. So this is a company that was founded together with Jennifer Doudna and Rachel Horowitz, who's now the CEO, back in 2012. And it's essentially evolving from a technology platform company into a company that can hopefully, and this is the kind of the mission is to really develop best-in-class next-generation CRISPR-Cas technologies to really focus on improving the specificity and then on its applications in cell engineering for therapeutic purposes. So things like engineered cells for immuno-oncology or engineering microbes to act as delivery vehicles to basically deliver therapeutics into patients. So the company is just over 5 years old. It's raised quite a bit of funding. We're now almost 50 we have now almost 50 employees, and things are going really well at the moment. So just to summarize what I've talked about, so I've shown you that this is a really powerful genome editing technology. it gives us really transformative capabilities, not just in basic science, but also for applications. Especially in somatic gene therapy, there's quite a lot of development, and quite a number of studies now show that this could really work quite well. We have now clinical trials ongoing for editing T cells for cancer immunotherapies, and so this is going to be, I think, this is going to continue, I think, bringing revolutionary advances in molecular medicine. Something that I haven't had time to go into is the potential of applying also these systems in germline genome modification, so in human germ cells or in human embryos, and this is currently highly controversial, and this is something that that basically warrants an ongoing discussion between the multiple stakeholders, scientists, policymakers, patient focus groups, legal and ethical experts, and so on, because it's something that we as humans will have to face up to at some point, whether it is or not a good idea to basically apply this technology in a way that can basically change the course of our evolution. So with this, I'll just spend the last few moments to acknowledge people in my group. So this is the team that I work with in Zurich. It's a fantastic group of people, PhDs, postdocs, technicians. They come from different corners of the world, and I've been really privileged to be able to work with them. We have a number of collaborations, both internally, but also in the Netherlands and elsewhere, New York, Berkeley, UC San Diego. So it's been an immensely exciting and fruitful time that I've had in the last 5 years, and I look forward to continuing working on this in the future. So thank you very much for listening. I'll be happy to take questions, and yeah, I hope I can talk to you. Do we do Q&A now, or
yes.
yeah?
If you want, you can just.
Okay, so yeah, we can.
I can talk. That's fine.
Yeah.
Is there a minimum length for the letters that need CRISPR or Cas9 to check DNA?
You mean?
Understand my question?
Yeah. So the RNA, the search string, so to say, is 20 letters, and so in theory this would give you enough specificity to target, a unique site in a human genome. So.
But in theory, it's possible to use less letters.
In theory, yes. So the system will be active with as few as about 16 or so. Yeah.
Thank you.
So if coming back to the origins of CRISPR, if the bacterium takes a part of the viral DNA into its own genome in order to then produce the guide RNAs, how come Cas9 doesn't cut its own genome then?
Yeah.
How come bacteria don't have autoimmunity?
Yeah. So the reason for it is that there's a kind of a there's a mechanism that specifically avoids that. Something I didn't have the time to go into, but maybe I can go to the slide. The system basically, in addition to needing this interaction between the RNA and the DNA that's mediated by the guide RNA, it only works if that site is actually next to a specific sequence. And this is, it needs basically a short sequence motif that needs to be right next to the target site. This is known as PAM, P-A-M, protospace or adjacent motif, and without it, the system will actually not recognize the DNA as a target. And the reason why bacteria don't target themselves is because when they integrate the DNA from the invader, they integrate it in a way that this is actually missing in the genome. And so, in that way, even though the target site is in the bacterial chromosome, it's not visible to the system. So this is actually what this slide shows. So in the system that is most widely used, the target site has to have two guanines, two G letters basically immediately downstream of it. And the system actually specifically recognizes them by these interactions. And if you don't have these two Gs, it will not actually target the DNA. So we can talk later if this is.
Oh, that's clear. But I have may I ask a second question that relates to this patent war that was going on. Yeah. So I'm not sure what was the final outcome, because I know that the Broad Institute won some patents and not UC Berkeley. I would like to ask whether this affects Caribou Biosciences directly, or whether Caribou Biosciences is sort of anti-fragile in that context.
Well, Caribou Biosciences has licensed some of the IP that came out of UC Berkeley, which is now part of this dispute with the Broad Institute. But Caribou has, over the years, also developed its own IP, so they're not completely dependent, basically, on this. No, maybe.
Yes. I recently read an article about CRISPR-Cas, and there it was stated that the technology, talking about humans, might be facing the problem that the human people might be immune to it, as bacteria is extracted from also is present in humans. And so my question is, do you see this problem as well, and is there a way of solving it? And yes.
So yeah, there have been studies showing that, or suggesting that, that could detect basically antibodies specific against, particularly the Streptococcus pyogenes Cas9 protein, because Streptococcus is a naturally occurring human pathogen, so humans come into contact with it. I'm not sure whether this is going to be such a major issue in the end, because especially for the ex vivo therapeutic approaches, you do it on a system that essentially doesn't have an immune system, so to say, and by the time the cells are introduced back into the patient, or would be introduced back into the patient, there's no trace of the Cas9 protein anymore. So for these kinds of applications, I don't think that there would be an issue. There could potentially be issue for in vivo sort of direct delivery of these systems into patients, but this is something that has not been investigated in enough detail. If anything, that at least in other animals, it doesn't seem to be such a big problem in the end. So there are now genetically engineered mice that basically express Cas9 constitutively, essentially throughout the entire organism, and they don't seem to have a major problem. So I would say yes, we will have to look into this. We will have to treat this with caution, but I'm optimistic that this is not going to be such a big problem in the end.
Thank you.
Yeah.
My question relates to the previous one. So I heard that people are trying to find bacteria, like some kind of the same system, that have never been in contact with humans. Do you know anything about that?
Yeah, so this would be a way to basically potentially avoid that, right? That really work with systems that have not been in contact with humans. There's a lot of effort going into kind of data mining, genome mining to identify new systems. And yeah, there's quite a lot of groups sort of active in this area. Maybe here.
So when you want to insert a gene with CRISPR, you just add the template to the CRISPR components and hope that the cell insert is by this reparation.
Yeah, so that's kind of the essence of it, right? Now, there are, again, various ways in which you can deliver that piece of DNA that acts as a template. You know, this could be synthetic DNA. This can be also a virus that basically carries this cassette or this template as part of its genome. So, again, it depends on the specific cells, the specific organism that you work with.
Yeah, I have a question. Maybe it's more a political question. I don't know if they're still going to talk about the technology itself, but you spoke about caution and oversight of modification of germ cells. And my question is, how far are we away from uncontrolled or rogue actors getting into that potentially? And what are the dangers, really? Of course, in the media you hear that, okay, this is the next super Ebola delivery system or something like this.
Yeah, I mean, you know, it's of course something to be to bear in mind, to be concerned about, but I'm afraid I don't have a good answer for how far we are from somebody kind of abusing this and going rogue. I think, at least in the scientific community, there's pretty strong consensus on what can and what should not be done. There have been these global conventions and meetings organized by various learned societies and so on to recommend policies and to issue recommendations basically to governments. Sort of the current consensus is that the technology itself is not ready yet for really human germline applications. So I think on that front, I think we're probably okay. There's of course the question of, yes, is there somebody who could use this, you know, and sort of really go rogue and do something? But you know, if anything, you know, we've had a same, you know, or a very similar debate when reproductive cloning came along. You know, when Dolly the sheep was kind of the topic of the day, everybody was predicting, well, it's only a year or two before somebody does it in humans, and that didn't really happen in the end. So, because I think in part there was enough self-restraint in the community that you know, nobody was willing to risk basically their reputation or their their position basically to do something like this. And I think we'll see something similar in the CRISPR-Cas field.
Yeah, in the past decades we have seen like a steady increase like in the lifespan of humans, but in developed countries it's like also like incremental, like we have seen no big jumps. And so what I'm very curious about, like when we go like on the time period like a bit longer, not maybe 5 years but way longer, is it possible like that CRISPR-Cas like allows like also like the lifespan not to be increased by 2 years but maybe to 2,000 years, something that we haven't seen before?
Yeah, I mean, I think this is really pure speculation at this point. I think I think my answer would be is that we don't really know enough about how, you know, what controls longevity to actually be even able to start thinking about how to manipulate it genetically.
The starting point was a bit like an article which was like talking about a lobster, a specific kind of lobster, which is expected like to live as long as it does, as long as it has no accident, as the cell reproduction goes on and on and on. And so then like the question was like, can we take this like specific like and put it in like the human cells?
Yeah, well, I don't think it's going to be as simple as that. In part because I think the genetic basis of longevity is still something that's not really very well understood, and I think the systems, you know, are probably not completely sort of transplantable between organisms. But again, you know, I don't think I have a very good answer at this point.
Which strategies are there for the delivery to specify the delivery?
You mean into cells or into
in special tissues? If you want to have a special target, maybe.
So there are several different ideas. One is to basically engineer viruses that have certain preference for a specific cell type.
Use receptors.
Either by engineering the receptors or by essentially just evolving them kind of in vivo or in vitro, by essentially adapting them to have sort of increased tropism for a certain cell type. And this is something that has been done and shown to work. Not for all cell types, but there are certain cells and certain viruses that do like each other, and where you can have some preferential delivery. So that's one. The other would be to, again, try to develop things like nanoparticles or other delivery systems that could be coupled to engineered proteins that can recognize specific epitopes on cell surface and so on. So I think this is where most of the development is, I think, with regards to specificity. Yeah, maybe.
What's the most cost-efficient approach to cure a genetic illness? To cure it in an adult or in a germ cell after the illness is diagnosed, or even in a preventive approach before any illness is diagnosed, just to look for it,
Yeah, I mean that's actually quite a really complicated question, because at least on the germline front, there's not much you can do at the moment. At least not in Europe anyway, because here germline genome modification is not legally permitted. Right? The situation is somewhat different in the US, although again there are restrictions, and so that's something that is not something that I think is being really actively investigated at the moment. On the somatic front, so modifying somatic cells in a way that the person who basically receives those cells cannot pass that modification onto its offspring. There it will again depend on which type of cell is being modified and what the delivery will have to be. But I think the initial focus on ex vivo approaches will probably be the most feasible one, and probably the least costly, at least in the short run.
I have a question of maybe a little bit different nature. I mean, there are so many people doing researches, maybe also on topics like that. What do you think was it that made you able to do this breakthrough of the year 2015 to publish that paper? Just all those milestones that have been done in the centuries in the science. What is this? Maybe something that is there a difference between you and other researchers, or as example, Professor Dubna or Charpentier? Maybe what do you think?
That's a good question. I think there is a lot of factors, I think, that goes into this. I mean, of course, one thing is that nobody does their science in a vacuum. You always base your work on kind of the state of the art in the field, right? What is published by others, and sometimes there's a very long history of certain, in certain fields or certain questions that people have attempted to tackle from multiple directions, and sometimes well, but sometimes quite unsuccessfully. So that's one aspect of it. You never really operate in a vacuum. The other is that some of these discoveries were really, quite unexpected. At least my initial motivation in this project was really to work out how these nucleic acid targeting enzymes work. It became very, very clear very quickly that this could have really tremendous potential for applications, but at least in the beginning, that was not quite, the mindset that I went into this project with. I trained as a structural biologist, so I think very deeply about how molecules work, and I get really excited, when I see things like this. So for me that was, at least part of the motivation. So it's the curiosity and the kind of the basic research aspect of it. And then the last one is, quite frankly, being at the right place at the right time. There is an element of luck in these discoveries, and that's something that's very hard to control, very hard to describe. I think I was very, very privileged to be in a great academic environment where we were encouraged to think about our projects, to ask the right questions, to kind of think outside the box, so to say, where we had the freedom to explore ideas and kind of work with other people and exchange ideas, and be quite pragmatic about the way we worked. So that's again something that goes into this, and and the rest is just really hard work. It's 10% inspiration, 90% perspiration, as other people have said. So it's all of this, yeah. And the key is to be in the right environment where all of this is fostered and encouraged. So, thank you. Yeah.
I have a question to better understand the challenges that we're facing, or science is facing, with the targeting of in vivo cells.
Yeah.
To modify them. So is it rather a matter of hitting the right cells so that we can actually do the modification in the cells that we want to modify, or is it a matter of not hitting the wrong cells? Do you get that?
Yeah.
Is there potential damage that you can do to other cells?
Yeah. Well, I think it's more of the former. That you really, if you because the system itself, the cutting is actually quite efficient as long as you can deliver it, as long as you can deliver the components into the cells efficiently. And so the key is to deliver it into cells efficiently and to deliver it into the right cells. Not so much because if you delivered it into the other cells in the organism, it would cause damage, but you really want to focus on the cells where it matters the most. So it's more of the former rather than the latter. Maybe in the back you've been trying to ask a question.
Yeah, I would like to discuss the technical aspects a bit more. What do you think? What should be done to start a discussion more in society, and what has already been done?
So I already kind of touched up on this. So there have been various meetings. There was this global genome editing summit, and
but only in the scientific community.
Well, I would actually disagree with that. Yes, the scientists were an important part of it, but But there were quite a lot of stakeholders that took part in these discussions, and I think this is something that needs to continue. Basically, it's not for the scientists to decide this on their own. I mean, that's very clear that there will need to be input from other parts of the society, because this can have potentially the impact on all of us. Right? So in a way, there will have to be it will have to be a multi-stakeholder kind of situation where, in the end, it's the society, it's the the politicians and the decision makers who in the end make a decision. But I would hope that it's an informed decision, basically. Yeah.
After you realized the potential of your discovery, what were your first crucial steps in founding the company?
Founding the company is one thing, but the company has to have, something to work with, right? And so the other important aspect of this is to have access to the IP, basically. So yes, on one hand, there's the whole process of incorporating the company and kind of doing all the the legal side of it, and this is something that at least in the US doesn't actually take all that long. But what you really need to focus on is to what is going to be your strategy vis-a-vis, for example, the IP and so on. How will you be able to license the IP from let's say the university or some of the other companies or other entities in the field and so on. So this is I think by far the most important thing, at least in the beginning, and then of course finding investors.
Question is, what is the role of your technology transfer office in Berkeley? How did they negotiate the IP licensing, and did it take equity from your company? And maybe tell us something.
I mean, it's probably not. Well, I don't know all the details, and there's there are certain things that I'm not at liberty to talk about. But basically, the tech transfer offices at the universities play a very important role in this, right? Because on one hand, they basically facilitate all the filing of the IP and so on, and then if there are potential licensees, they handle the licensing process, and again, depending on the technology, depending on the potential, depending on what kind of a licensee it is, then the license can have different structures, right? Sometimes it's a sort of a cash deal. Other times it's more of a equity loaded deal, depending on what the technology is also. You want to follow up, I guess.
Another one.
Yeah.
I guess many investors were standing in the queue to get a part of your company and to invest in your company. So 41 million is quite money. Could you choose among 100 investors, and did they come to you, or did you have to find them? And did the tech transfer office help you with it?
So in the beginning, it was not straightforward. Actually, this is the early phase is convincing. Somebody other than your friends and family to give you money is I think the hardest. And then the tech transfer office was actually not involved in that, but the company actually started its existence in one of these incubator type bodies. So the University of California has an incubator basically, and they could essentially rent both lab space and office space for quite a convenient and good rate, and there was support basically on that front, and so these kinds of activities are actually quite important, and in the case of Caribou they were actually quite crucial in the beginning.
When you have the CRISPR-Cas9 in your cells and in your body, will they be there your whole life?
Well, it depends on how it's delivered and how what the actual method is. Okay, so in some cases, and I'm not talking about humans here. In some organisms or some cell types, you can basically deliver the system using a virus that inserts its DNA into the DNA of the host, and in that case, the gene encoding the protein will be fixed in the cells that have been infected with this virus. There are other systems like the recombinant protein RNA approach, where basically you're delivering just the protein and the RNA as a complex, as a mixture, and these components actually get degraded in the cells over time. And so with this approach, you basically 24 hours later, there's no trace of the protein, there's no trace of the DNA, because and since there was no DNA delivered into the cells that would encode these components, there's no permanent record. So this is actually something that is actually also very good for the potential therapeutic applications, because you really you deliver, it has its effect early on, and then at some point it will get degraded.
When is this point? How long does it take?
About 24 hours. I mean, it again depends on the cell, depends how much you deliver to begin with, what the effective concentration is, and so on. But studies have shown that within about 24 hours, and we see this also in our experiments, the levels go down, and at some point you just don't detect the protein anymore. And
I have two more questions from the online community, because not everybody could come and sit inside, and they have an ethical background, and it's like the first one is, which modification through CRISPR would you say are worth striving for, and which aren't? And the second question is, do you think that people becoming parents should be able to decide what kind of child they want?
So the first one, what modifications are good and what would not be so good. Well, I think we can probably all agree that treating genetic diseases is probably a good thing. I would argue specifically, using the somatic cell approach, there's, from at least my point of view, ethically, there's no problem. For the germline genome editing, I think currently, given that the technology is still not quite ready to be used in that kind of context, and in the society there isn't a strong consensus for the use of it in this way, then I would say, well, it's probably not worth going ahead. One thing that I feel quite strongly about is also potential non-therapeutic applications, and this is something that I would probably not encourage at this point.
You are referring to designer babies.
So things like basically making modifications that are not there for the purpose of curing a genetic disease.
Yeah. But do you think that then also the definition of what is healthy and what is not healthy,
right?
Will also
and that's something that may evolve over time. Yeah. So I think I think this is something that will continue, driving debates for a long time.
Okay, but maybe also like to precise like what we can take from this evening, like the application on like designer babies is not feasible at the moment, right?
But at this point, this is really not, it's not realistic, basically, to even contemplate something like this. In part because for many of the things that, are sometimes thrown out there as potential, targets for modification, like intelligence or eye color, hair color, body dimensions, all these things. These are all multifactorial, multi-gene traits where the contributions of individual genes are not always easy to discern, and so
it's more complex than to treat Down syndrome, for example.
Well, that again is something that would probably not be possible to treat using this approach. Right now, we're really talking about applications where there's a single well-defined gene that has a well-defined mutation or some kind of modification that has really direct consequence for the disease. Those will be probably treatable within my lifetime, I would say. Yeah.
So I think there are still a couple of questions, but we would like to kind of open the whole ensemble here. And we have a buffet, for example, and then we can just talk personally with Professor Jinek and everything in just a little bit cooler way. First of all, I just want to thank you very much, to thank you for your time and to come here. I think there may be some controversial points, but it's always like that if you're working on something great and something that has a great impact on the whole world, as for instance, and still, we thank you that you came here, and we have a little kind of present for you.
Okay.
And yeah, I hope you can just enjoy the evening a little bit with us.
Okay, thank you very much.
No problem.
It's really appreciated. And thank you for the invitation. Yeah.